Hello students, welcome to the session. Myself Shobha from Maharani Science College, Mysore. Today, I am here to discuss about the topic glycogen metabolism, which is also an important biochemical pathway in carbohydrate metabolism. You know that when we use the term metabolism, it includes both anabolism and catabolism, right? Hence, today I am going to talk about two concepts. One is glycogenesis, which is an anabolic pathway and is nothing but the synthesis of glycogen. And the second concept is glycogenolysis, which is a catabolic pathway, or here the breakdown of glycogen takes place. Okay, now let's move on to the synthesis of glycogen. But before that, I would like to refresh your knowledge about the structure of glycogen. You know that the glycogen is a homopolysaccharide made up of glucose units. It is a branched chain polymer which consists of a linear chain of glucose linked via the alpha-1,4 linkage, and for every 8 to 10 glucose residues, a branch point is seen at alpha-1,6 glycosidic linkage. And this polymer also has two ends. The end which has free aldehyde or a hemiacetal group at C1 is the reducing end, and the other one is the non-reducing end. And coming to the functions of glycogen, this is the primary storage reserve of glucose and is mainly stored in liver and the skeletal muscle. So continuing with the functions of glycogen, the glycogen which is stored in the liver helps to maintain the sugar level in the blood during early stages of fasting, or you can say when there is a long gap between the meals. Whereas the glycogen that is stored in the muscle, it serves as a reserve of glucose for ATP synthesis during strenuous exercise. Now let us look at this liver glycogen, where it makes up around 8 to 10% of the fresh weight of the liver. Main function is to maintain the blood glucose level between the meals and hence depleted after 12 to 18 hours of fasting. Coming to the muscle glycogen, it makes up 2% of the wet weight of the muscle, and main function is to supply the glucose for the muscle during contraction, and this is depleted after prolonged or a strenuous exercise. Okay, now when we are talking about glycogen, an important question to be addressed here is why polymers, which means what is the purpose of storing glucose as glycogen? I know you will definitely answer glycogen is a glucose reserve which can be used later. But my question here is why glucose is not stored as glucose itself, but it is stored as glycogen. This is because if the cell store excess of glucose inside, it will create the osmotic problem. Okay, let me explain you this how. You know, osmosis is a colligative property which depends on the number of solute particles. Now, if more number of glucose is stored in the monomeric form, it will definitely increase the solute concentration inside the cell, and thus the solvent from outside start entering the cells, what we call as end osmosis. So, the cell will burst. In order to prevent this osmotic pressure, the glucose is stored in its polymeric form as glycogen. Hence, storing glucose as glycogen is not just advantageous as a reserve of glucose, but it is also an essential process. So in order to avoid the osmotic pressure, the monomers are not stored, but it is converted to its polymeric form. Okay, next important question about glycogen is why glycogen has more branching. This is because the branching in the glycogen increases the solubility. Without branching, the glycogen will precipitate inside the cell and this leads to a glycogen storage disease type 4, which is actually an inborn error of metabolism. And the second point is the branches creates more of non-reducing ends and that are available for both polymerization and for the breakdown. So let me explain you this. Why non-reducing ends are important? This is because during glycogen synthesis, glucose is added to the non-reducing end for the chain growth, and even during hydrolysis or during the breakdown of glycogen also, the glucose is released from the non-reducing end so that more the non-reducing end, more number of glucose can be released simultaneously, or you can say the process will be rapid. Hence, glycogen has more branching. So, one is it increases the solubility, and the second point is branching creates more non-reducing end that helps it for both polymerization and also for the breakdown of glycogen. Okay, now let us know when glycogenesis takes place or when does the synthesis of glycogen takes place. So you know it is when enough glucose and energy is available in the cell, it triggers the synthesis of glycogen. So here you can see glucose once when it enters the cell, either liver cell or other cells by specific transporters, it is immediately phosphorylated to prevent its diffusion outside the cell. And this glucose 6-phosphate which is formed, depending on the need of the cell, it can either enter glycolysis or HMP shunt, or when excess of glucose and ATP is available, it is stimulated for its storage via the glycogenesis or by synthesizing the glycogen. So students, now you know what is glycogenesis, where it takes place, what is its importance, right? Now let us move on to the reactions of glycogenesis. Here you know, once the glucose has entered the cell, it is phosphorylated to form glucose 6-phosphate, and this glucose 6-phosphate is isomerized to form glucose 1-phosphate catalyzed by the enzyme phosphoglucomutase. Let me explain you this reaction using the structures. Here you can see the glucose 6-phosphate is converted to glucose 1-phosphate by the enzyme phosphoglucomutase. Here I would like to tell you about the mutases. These are the class of isomerases involved in catalyzing the intramolecular group transfer. So they transfer the functional group from one position to another position. Here in this reaction only you can see the phosphate group present on the 6th carbon is transferred to the 1st carbon, forming glucose 1-phosphate. The next step in the glycogenesis is activation of glucose, where the synthesis of uridine diphosphoglucose or UDP-glucose takes place, catalyzed by the enzyme UDP-Glucose Pyrophosphorylase. This reaction is actually a committed step of this pathway and is an irreversible step. This is because here the glucose is activated using a high energy nucleotide UTP, forming UDP-glucose. Okay, now it is time to remember a scientist, Louis F. Leloir, for his discovery. See, the metabolic role of this nucleotide sugar UDP-glucose was discovered by him, so for which he was awarded Nobel Prize in Chemistry in the year 1970. So let me continue with the glycogen synthesis, where in the second step, UDP-glucose is synthesized, or activation of glucose we call this as, where glucose 1-phosphate formed in the previous step is activated to form UDP-glucose by utilizing the high energy bonds of UTP. So there are two terminal phosphates of UDP is hydrolyzed and is lost as pyrophosphate, thus forming the UDP glucose. That is why, as I told you, this step is the committed step of the pathway because the activated sugar is formed at the cost of two high energy bonds of UTP. You know, UTP is the energy equivalent of ATP. So till now we have discussed these reaction steps of the glycogenesis. So starting from glucose, which is phosphorylated to form glucose 6-phosphate, and glucose 6-phosphate which is isomerized to form glucose 1-phosphate, and now this glucose 1-phosphate is activated to form UDP glucose. So once the UDP glucose is formed, the next step is nothing but polymerization, or the chain lengthening process, which is catalyzed by the enzyme glycogen synthase. So the UDP glucose function is it transfers the glucose, the activated glucose residue, to the existing glycogen chain so that the glycogen grows in length. So here you can see the structures to understand the reaction. The UDP glucose, the C1 of the UDP glucose is transferred to the C4 of the glycogen chain, thus forming alpha 1,4 glycosidic linkage, and this step is catalyzed by the enzyme glycogen synthase. As you noticed in the previous step, the enzyme glycogen synthase adds the glucose residues from the UDP glucose to the existing glycogen chain, which means a primer is required for the synthesis of glycogen. So a fragment of glycogen can serve as a primer in the cells where glycogen stores are not totally depleted, but in case if the glycogen fragment is not present, which means if the entire glycogen store is utilized or depleted of, then a protein called glycogenin, which has a glycosyl transferase activity, serves as the primer. So now you know glycogenin can act as a primer and thus it initiates the glycogenesis. It accepts the first glucose residue from the UDP glucose to its own tyrosine residue, forming O-linked glucose residue. Then the glycogenin catalyzes the transfer of another glucose from the UDP glucose to the carbon 4 of the attached glucose. So it forms an alpha 1,4 linkage. This process is repeated until the glycogenin synthesizes a linear chain of around 8 glucose residues, which are all joined by alpha 1,4 linkage. Then the glycogen synthase continues the polymerization to form a chain of 10 to 12 glucose residues. Next step of glycogenesis is the branch formation. When the glycogen synthase has formed a linear chain of 10 or more glucose residue, another enzyme called as amylo 1,4 to 1,6 transglycosylase, or we can simply call it as a branching enzyme. This hydrolyzes a terminal segment of at least 4 glucose residue from that linear chain and inserts it with an alpha 1,6 glycosidic bond, and thus it forms the branch point in the glycogen. And you remember, the branching increases the solubility of the glycogen inside the cell, and also it increases the number of non-reducing ends which are required for both polymerization as well as for the degradation. And this is how, by the action of glycogen synthase and by the branching enzyme, a glycogen, a highly branched storage polysaccharide in animals, is formed, which has a diameter of around 20 to 50 nanometer and can have up to 120,000 glucose residues, and it is stored as granules in the cytosol. This is the glycogenesis pathway what we have discussed so far. Glycogenesis is triggered whenever there is an excess of glucose and energy available. So this glucose inside the cell is phosphorylated to glucose 6-phosphate, and which is further isomerized to form glucose 1-phosphate. And this glucose 1-phosphate, by making use of an UTP, forms an activated form of glucose called UDP-Glucose. And UDP-Glucose starts transferring the glucose residue for the growing glycogen chain by the action of glycogen synthase. But this glycogen synthase, it requires a pre-existing fragment of glycogen. If it is not available, then the protein glycogenin can act as a primer in the absence of a pre-existing fragment of glycogen and can form alpha-1,4 glycosidic linkage. Then comes the branching enzyme, which introduces the branch point at alpha-1,6 glycosidic bond. So this is how the glycogen is synthesized in the liver and the muscle cells. So now let us move on to the second concept under glycogen metabolism, that is glycogenolysis. If you remember, in the beginning of the session I told you, under glycogen metabolism we are going to discuss about two concepts. One is glycogenesis and the other one is glycogenolysis. Now I hope you know about the glycogenesis, which is nothing but the synthesis of glycogen and is an anabolic pathway, whereas this glycogenolysis is a biochemical pathway in which the glycogen breaks down to release the glucose molecules. Hence it is a catabolic pathway. In fact, this is how the stored glycogen in the body is utilized, because glucose is a source of energy for the body. So during fasting state, in order to maintain the normal blood glucose level, liver plays a central role in producing the glucose by this process of glycogenolysis, where glycogen undergoes degradation to release glucose and also the energy. Okay, now if we look at the reactions of glycogenolysis, the pathway begins with the action of the enzyme glycogen phosphorylase that releases glucose as glucose 1-phosphate from the non-reducing end of the glycogen. See, through the structures I think you can understand it. See here, if you can look at the structure, the glucose from the non-reducing end of the glycogen is released as glucose 1-phosphate, and now the glycogen is shortened by 1 glucose residue. And another thing you have to observe here is, for the phosphorylation of glucose into glucose 1-phosphate, instead of ATP, the enzyme is utilizing an orthophosphate. Can you tell me why? This is because the glycogenolysis is a reaction which takes place when there is a low energy state of the cell. So, in order to spare any available ATP which is required for other important processes of the cell, ATP is not utilized here for the phosphorylation, but instead an orthophosphate is utilized to remove glucose as glucose 1-phosphate. This action of the enzyme phosphorylase is repeated in the non-reducing end to release glucose as glucose 1-phosphate, but it stops when it reaches the glucose residue which is 4 units away from the branch point. This is because the enzyme phosphorylase can only act on the alpha-1,4 linkage and not on the alpha-1,6 linkage. So here comes another protein with two enzymatic activity. One is transferase activity and the other one is the debranching activity. The transferase activity of the enzyme transfers 3 out of the 4 glucose residues from the branch point to the linear chain or to the main chain, leaving only 1 glucose attached to the branch point. And here comes the debranching enzyme, which is also called as alpha-1,6 glucosidase, that removes the glucose from the branch point, hydrolyzing the alpha-1,6 glycosidic linkage. And then as usual, phosphorylase enzyme continues cleaving alpha-1,4 bond from the main chain, and thus by the action of phosphorylase and debranching enzyme, glycogen is completely degraded to release glucose molecules. So this is about the glycogenolysis, where the glycogen phosphorylase enzyme removes glucose from glycogen as glucose 1-phosphate, and this glucose 1-phosphate formed can be isomerized by the enzyme phosphoglucomutase to form glucose 6-phosphate. But this glucose 6-phosphate can be utilized by the cells depending on its need. It can either enter muscle and brain where it is completely oxidized to pyruvate and also to lactate, or in the liver cell it can be dephosphorylated to form glucose and released into the blood for its use by other tissues, as well as it is used to maintain the blood glucose level. Or if there is a requirement of the ribose sugar and NADPH, the glucose 6-phosphate can also be channeled to the pentose phosphate pathway. So to summarize the importance of this pathway, glycogenolysis takes place in the muscle and liver tissues in response to the hormonal and also to the neural signals. It plays an important role in the fight or flight response and in the regulation of glucose level. In the muscle cells, it provides an immediate source of glucose 6-phosphate for the glycolysis to provide energy for the muscle contraction, whereas in the liver cells, the release of glucose into the bloodstream for uptake by other cells as well as to maintain the blood glucose level. So this is how the stored glycogen in the body is utilized in times of need by the process of glycogenolysis. Thank you.